Hi, today I'm gonna go over DNA and DNA replication. It's the first few slides of this big idea PowerPoint. Um, and although we already know what DNA is, I might as well just go over it. And in this PowerPoint, I skipped the first few slides because I didn't think they were that relevant, and it's just like by some facts that you could learn by yourself. Okay, so in DNA, there is a phospho um, phosphate backbone, and then there's a bunch of sugars that are connected to the backbone. The backbone's actually phosphor sugar, and there are nucleotide bases attached to the sugars, and those sugars can be four letters: A, T, C, or G. A matches up with T, C matches up with G. DNA is anti-parallel. That means it runs at opposite ends. This side goes from 3 to 5, and this side goes from 5 to 3. And of course, you know that it makes a double helix because that's pretty common knowledge. And again, the RNA is very similar to DNA. It also has a phosphosugar backbone, and it also has a bunch of nucleotide bases stringing from it, but it's only one strand. See, DNA is two strands here that are intertwined with each other. RNA is only one. And instead of T, RNA has U. Okay, now we're going to get into DNA replication. Since the two strands of DNA are complementary, which means they're opposite each other, each strand acts as a template for building a new strand in replication. So the strands split apart, and each of them are a template for a new strand of DNA, and since opposites match, you'll get two of the same DNA from one DNA. And again, just a summary, in DNA replication, the parent molecule unwinds, and two daughter strands are built based on base pairing rules. This is a really good, simple picture of DNA replication. This is the parent molecule. It separates. Then new nucleotides and phosphate backbones attach, complementary, and you see we have two of the same exact DNA that we started with. And those are the three major phases of DNA replication: unwinding, base pairing. No, it's initiation, unwinding, base pairing. And then, okay, replication begins at particular sites called origins of replication, where the two DNA strands are separated, opening up a replication bubble. So for replication to start, then there's the first place where DNA strands are separated, and that's where it starts. Um, this is not that important. Okay, this is the first step, unwinding. Helicase, which is enzyme that you should know, unwinds the DNA, and that's the first step. And while that's happening, for DNA to start attaching to it, to the for new DNA to start attaching to the old DNA, first they need like a little boost, and that's called the RNA primer. So primase makes the RNA primer, and that's just like the little template that it needs for the DNA to start attaching. Just a summary. At the end of each replication bubble is a fork, which is a Y-shaped region. This Y-shaped region where it's elongating so it could separate. Helicase separates it. Binding proteins are these little gray things that are binding to the DNA that help it, um, that help stabilize it, and this enzyme topoimerase affects overwinding and breaking and swiveling and rejoining. And this is really important. DNA polymerase, which is the enzyme that actually adds the new bases, cannot initiate synthesis of a polynucleotide. They can only add nucleotides to the 3N. So that's why you need an RNA primer to start it, and then the DNA polymerase just starts adding on to the 3N, and it just starts adding bases. But you need this RNA primer for it to start. Um, and I'm just going to show you this with a picture, but DNA, let's say in this picture, this DNA is being unwinded that way. So this side is closing, 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 and this side's opening, opening, opening. The top strand, since DNA can only add to the three direction, this has no problem. It just goes in the direction that it's unwinding. But in the bottom strand, which is called the lagging strand, DNA is being unwinded here and it's closing here, but it has to add bases this way because this is the three direction. So if it has to add bases this way, but DNA is unwinding this way, it adds bases until it reaches where it's closed. It adds bases until it reaches where it's closing, goes back, adds more, goes back, adds more, goes back, adds more, in like a little backwards circle type of motion, as you can see in greater um, detail here. That's a primer. It adds DNA, but it reaches where the DNA is closing, so it has to go back, and the RNA starts again, and the DNA connects to the next primer, and it just keeps on going backwards and backwards. And each of these fragments are called Okazaki fragments. I just want to check if I forgot any. Okay. And DNA is all these fragments have to be connected somehow. So DNA ligase, which is this blue thing, blue or yellow, it's hard to tell, um, is the glue that holds them together. And again, DNA ligase joins the fragments. And I already explained this, so you could rewind in the video if you need to do this, but I'll explain it like simply again. DNA unwinds because from helicase, which is this green one, unwinding it. Then an RNA primer starts, and then DNA polymerase just keeps on adding, and the leading strand, which is the top strand, just keeps on adding, and doesn't have to go back because the DNA is unwinding in the direction that it adds. But the DNA has to add in the lagging strand, which is the bottom strand, in the opposite way to the right, even though DNA is unwinding this way. So it adds bases, goes back, adds more, goes back, adds more, goes back, adds more, goes back, adds more in the direction my mouse was moving. And of course, there's mistakes made when the polymerase adds up pairs. So they have to proofread the DNA. In mismatch repair, repair enzymes correct errors. In nucleotide excision repair, a nuclease cuts out and replaces damaged stretches. So this is the nucleotide excision repair. Nuclease is taking out a whole section of DNA, and DNA polymerase is putting new in, and DNA ligase glues it all together. Oh, I'm not that good at explaining this, telomeres, but I'll try my best. The ends of each gene have telomeres, which are special nucleotide sequences. It's typically TTAGGG, and that's typically repeated a bunch of times. Telomeres protect genes from being eroded through multiple rounds of DNA replication. So you have all these primers, right? When you're in the lagging strand, because you have to keep on going back and starting again. So there's a bunch of RNA primers, and when you remove those primers, DNA can replace all the primers except the last one. And so um, when the RNA is removed at the end and all the DNA that can be replaced is replaced, the leading strand has the perfect amount and it's a perfect DNA model, but the lagging strand has extra DNA at the side. And that leads to shorter and shorter daughter molecules, but that's okay because at the ends there's telomeres, which are basically just useless, not useless, but like just sequences of DNA that have no function. So you're basically just sacrificing the part that has no function. See, um, this is the lagging strand, and once the primers are all removed, there's a missing part, but that's okay because it's a telomere. And telomerase uses a short molecule of RNA as a template to extend the three end of the telomere. So just to prevent from further erosion, it adds to this end on both sides of the DNA molecule. Now there's room for primase and DNA polymerase to extend the 5' end because when it copies it, it has to match it with the 3' end. So if it extends this one, then it can extend that one. Does that make sense? Sorry, I said it was bad at explaining that. And that's pretty much